We will talk about the cardiac cycle, conduction system of the heart, some basics of the blood vessels now. So this here is the beginning of the cardiac cycle. The heart is completely relaxed and the blood freely flows into the ventricles. This time is called diastole. At some point, atria start to contract. It is called atrial systole, and the blood is being squeezed into the corresponding ventricles through the atrioventricular valves, either tricuspid or mitral. As the result of this atrial systole, pressure in the ventricles rises and ventricles start to contract. So this is the ventricular systole. Okay. Ventricles start to contract, pressure elevates even more, and that elevated pressure closes atrioventricular valves, either tricuspid or mitral. Pressure in the ventricles as the result of the ventricular systole keeps going up, and that pushes the blood into the pulmonary trunk, if we're talking about the right ventricle, or aorta, if we're talking about left ventricle, through the semilunar valves, either pulmonary or aortic. And pressure in the pulmonary trunk or aorta, as the result of it, rises, and this elevated pressure will lead to the closure of the pulmonary or aortic valves. Now, pressure that is developed during the contraction of the heart during the systole is called a systolic pressure. For instance, if your pressure is measured 120 over 80, 120 is the systolic pressure, and pressure in the aorta during relaxation is diastolic pressure, that's 80. Okay, so that's the pressure that we can measure in the systemic circuit during systole, and that is the pressure that we can measure in the system during diastole. So now we're going to move to our conversation to the discussion of the intrinsic controls of the heart. Important feature of the heart is that it can contract without any external nervous stimulation due to the presence of so-called pacemaker cells, cells that set up the contraction rhythm. The set of pacemaker cells, called the intrinsic control system of the heart, the conduction system of the heart, establishes an electrical flow through the cardiac muscle. It consists of several elements, and the first element is here is sinoatrial node. Sinoatrial node is it sets the actual normal healthy heart rate. Sinoatrial node is located in the right upper atrial wall, and it sends the electrical wave across the entire atrial myocardium. As the result of it, cardiac muscles in the atria depolarize and atria contract. Signal that is sent by the SA node reaches second element, AV node, located between atria and ventricles, so-called atrioventricular node, and the signal the electrical signal is being delayed at the atrioventricular node. It's really important that it's delayed because that allows atria to contract first before ventricles. Signal goes through the atrioventricular node and enters atrioventricular bundle. You can see the bundle here. And from the bundle, signal goes to the Purkinje fibers. So the function of the atrioventricular bundle and the Purkinje fibers is to deliver electrical impulses through the interventricular septum to the walls of the ventricles, and therefore initiate the ventricular contraction. It is important to highlight that these intrinsic controls also have another level of control from the rest of the body. So heart can contract without any nervous stimulation, but essentially nervous stimulation can change the heart rate, basically may override the intrinsic controls, and the signals from the cardiac center in the medulla oblongata can either increase or decrease the heart rate as necessary. But essentially, sinoatrial node is the chief structure that sets up the heart rate. The activity of intrinsic conduction system of the heart can be measured in the EKG. Essentially, EKG or ECG, electrocardiogram, measures the electrical current through the heart. It consists of several elements. First of these elements is a P wave. P wave shows the firing of the pacemaker cells at the SA node, and it signifies atrial depolarization and atrial contraction. When ventricles depolarize and contract, that moment is characterized by the so-called QRS complex. And when ventricles relax, when ventricles repolarize, you can observe T wave on the EKG. There are a couple of interesting elements here. One is so-called PR interval. You see it's the distance between the P wave and the R of the QRS complex. That's the time that it takes for an electrical signal to go from atrial depolarization in atria to ventricular depolarization in ventricles. And QT interval is the total time of ventricular contraction. The longer the QT interval, the longer your heart is squeezing the blood out. Now, what if there is a missing wave? What if you do not have a P wave on the EKG? There's no P wave on the EKG. It means that your atria do not contract. SA node doesn't work. If you see P wave but no QRS and no T wave, it means that for some reason AV node did not allow the signal through the ventricles, and ventricles did not contract. Atria did, but ventricles didn't. Now that brings us to the conversation about the blood vessels. So there are three types of blood vessels: arteries, capillaries, and veins. So in both arteries and veins, you can see three layers, so-called three tunics: tunica intima, tunica media, and tunica externa. Tunica intima, or the inner layer, consists of endothelial cells, the epithelial lining of the blood vessels. The middle layer, tunica media, consists of smooth muscles and elastic tissue, and connective tissue provides structural support in the outer layer, or tunica externa. So the main difference between these blood vessels is that arteries carry the blood away from the heart, and they originate on the ventricular side. Now, veins bring blood back to the heart. They usually have thinner walls, and they end up on the atrial side of the heart. They delivered to the atrial side. There are certain significant differences between arteries and veins. Arteries have thicker walls than veins. The closer to the heart the arteries are, the bigger they are. They're pretty large. They have thick walls. With each heartbeat, they stretch and they recoil. And if you move away through your sort of arterial tree, away from the heart to smaller arteries, they're going to become narrower, smaller. Wall will become not as thick, but they will be able to effectively dilate and constrict. Veins are not that good in terms of the dilation and constriction. But they are very, very stretchable. They have low pressure, and because of that low pressure, veins do not really need thick walls. You can see that the walls of the veins are relatively thin. Blood still flows through the veins quite effectively despite of the low pressure, because there's not much friction due to the endothelial lining. And now, between the arteries and veins lie capillaries. So here you can see arterioles. They lead to the capillaries. And capillaries are smallest blood vessels. They all consist of endothelium and basement membrane. You can see there's no smooth muscle. Very thin walls, practically single layer of cells. Gases, nutrients can go back and forth between the blood and the tissue. We call it a capillary bed, the set of capillaries in a certain tissue, and that's where all exchange of gases and chemicals occurs. So basically, arteries take the blood away from the heart towards the capillaries. Arteries become smaller and smaller, delivering the blood to the capillary bed. In capillary bed, there is an active exchange of the fluids, and then blood is collected from the capillary bed into the venules. Smaller venules merge, forming larger veins, and they merge, forming even bigger veins, which eventually bring blood back to the heart. Now that defines two circulatory circuits. One circuit is pulmonary circuit. We mentioned it already. It takes blood to the lungs. And another circuit is the systemic. So essentially, pulmonary circuit takes the blood, deoxygenated blood from the heart, delivers it to the lungs, where blood gets oxygenated, and then pulmonary circuit returns the oxygenated blood to the left side of the heart. And left side of the heart delivers oxygenated blood into the systemic circuit, where blood drops, kind of, quote unquote, drops off oxygen and nutrients in the tissues, and takes away wastes and carbon dioxide and brings it back to the right side of the heart, and the whole cycle starts again. So obviously systemic circuit is much bigger, so it has high resistance and higher blood pressure. But what's interesting, both circuits carry the same amount of blood despite the significant size difference. That actually explains why the blood flow in pulmonary circuit is much slower than it is in the systemic. Now finally, before we wrap up that particular chapter, portal systems in the heart, in the circulation, we mentioned before that in the majority of cases, you have arteries that deliver the blood to the capillary bed and veins that drain the capillary bed. But here you can see an interesting feature right here. It's called hepatic portal system. So this is the portal vein, and you see that it connects one capillary bed that collects the nutrients from the intestine and takes the blood, nutrient-rich blood, into the liver capillaries. So here you see two capillary beds are connected by the vein. So we call this hepatic portal system. So in the portal system, two capillary beds are connected by veins. So it's not just arteries, capillaries, veins, but it's going to be arteries, capillaries, veins, capillaries, veins. Okay? So there's more than one portal system. We're not going to chat about others, but I just want you to appreciate what the portal system is. The portal system in the liver connects the intestinal capillaries and capillaries in the liver. That allows to deliver nutrients from intestine to the liver for processing, and also to possibly clean the detrimental compounds that were accidentally picked up in the digestive capillaries.